The third research method is the experiment. Many researchers consider it the gold standard because it allows us to make causal inferences about the data we collect, unlike descriptive and correlational methods. Experiments answer why questions. Why do some people like working in groups, while others despise it? Why do some teams perform better than others? Why do some people resist change at work, while others embrace it? This method requires 4 key components: random assignment of participants, manipulation of an independent variable, the IV, control, and measurement of a dependent variable, the DV. The first step of an experiment is to create 2 or more equal groups using random assignment. After participants give their informed consent, they're randomly assigned to either the treatment group or the control group. This means each person has an equal chance of being in any one of the groups. They might be in the one that receives special training or in the one that doesn't. It makes it possible for us to assume, with at least some certainty, that the groups are similar in terms of their gender, age, ethnicity, education, attitudes, values, skills, and so on. This randomness reduces the likelihood that individual differences contribute to any observed differences. However, it doesn't mean the groups are exactly the same. They may differ in some other way. It's just one attempt to limit the influence of variables other than the ones we're studying. The next requirements are manipulation of the IV and measurement of the DV. In some way, we treat the groups of participants differently. For example, one group completes an easy task, the second group a difficult one, and the third group skips the task altogether. This treatment is called the independent variable, the IV. Then we measure participants' responses, which are called the dependent variable, the DV. So, after the easy task and difficult task groups finish their work, we measure everyone's stress levels, even the control group. To summarize, we manipulate the IV to measure its effect on the DV. In addition, it's common to measure what are called subject variables, which cannot be manipulated by the researchers, such as gender, age, and ethnicity. These variables are useful for statistical analyses, allowing us to better understand whether the IV has the same effect on the DV for different groups of people. For instance, do older people have the same experience as younger people? Or do people from Western cultures have the same experience as those from Eastern cultures? The final requirement is that we control for confounding variables. These are extraneous variables that could have influenced the results, but are not the main variables of interest. To address this issue, we can use a control group, treating it the same as the treatment group in every way, except it doesn't receive the treatment. By limiting the influence of extraneous variables and treating equal groups differently, we can be more certain that any observed differences were caused by the IV and not some other variable. Then, to determine whether a measured difference is of any significance, we calculate the likelihood that it occurred by random chance. The observed difference is said to be significant if the probability of it happening by random chance is less than 5 out of 100. So, the main advantage of the experimental method is that it has the most inferential power, enabling us to draw conclusions about causation. However, this method has a few disadvantages. First, no matter how hard we try, it's impossible to control every single variable with complete certainty. There's always a chance the participants were influenced by another variable, one we didn't notice, measure, or control for. Even the researcher's presence can influence participants. Experimenter expectancy effects occur when the researcher's behavior is influenced by their expectations of the study, and then they unintentionally influence the participants, and thus the results. To minimize these effects, we can use a blind study and avoid knowing which group each participant has been assigned to. It's also impossible to perfectly mimic the real world, and so this method varies in its experimental realism. Participants may not act naturally if they are being observed, as the researcher's presence alone is enough to impact how they think and act. Field experiments tend to be more realistic, but have less control than laboratory experiments. Let's return to Zuri and Rateau's study of the black sheep effect for an example. Participants in the study were randomly assigned to one of 8 groups in a 2-by-2-by-2 design. The independent variables were group identification and type of opinion. Group identification was manipulated by changing the name, and thus the assumed gender, of the fictional target. The researchers manipulated 2 types of opinion. One was changing whether the target's opinion aligned with the majority of the group, or ran counter to it, and the other was by changing whether the group's central or peripheral opinion was the focus of the investigation. The dependent variable was the participants' evaluations of the target. Essentially, how were their impressions of the target influenced by the details they read about them? What happens to their evaluations if the target is an insider and doesn't agree with the group? Were they evaluated more harshly than an outsider who doesn't agree? As you can see, these researchers investigated several questions within a single experimental study, which is common in the social sciences. Note that if a study does not have all four of these components, it's not a true experiment. Instead, it's called a quasi-experiment.